With the increase in global urbanization, many people are beginning to lose their connection with nature. And as our worlds drift further apart, we not only forget our crucial role as key stewards of this earth, but we also forget how nature affects our physical and mental well-being. In the Atherton tablelands, excessive land clearing for agricultural activity has led to fragmentation of the forests that used to populate this broad region of the wet tropics. Therefore, every remaining fragment of rainforest is important, no matter its area or relative age. By protecting local pockets of biodiversity, we protect wet tropics biodiversity as a whole, and through measuring it, we can assess the health of specific ecosystems and foster meaningful connections between people and nature. These connections are what will drive the future of conservation.
Our directed research occurred at the perfect time because we got to experience the very first BioBlitz in the region, thanks to the efforts of Gemma Horner.
So a BioBlitz is an intensive all-day or for a discrete period of time scientific study which encourages the local community to join with local experts, scientists and researchers to collect biodiversity data over a specific area.
The Malanda BioBlitz was held in Ngadjon-Jii Park, a revegetated public park in the small town of Malanda.
It was a very unknown area. To me, just from a preliminary look, had a reasonable amount of biodiversity, but was also disturbed, so that was interesting to be able to see what species are here and its proximity to the state school.
Knowledge and appreciation of nature is crucial to the future of conservation. When people are given the tools and opportunities to interact with their local biodiversity, they're more likely to care about protecting it. A large part of the BioBlitz focused on iNaturalist, a software designed to empower citizen scientists. Anyone can be a citizen scientist by uploading pictures of organisms, which are then ID'd and reviewed by a broader community.
Our BioBlitz team arrived the night before to kick off the 24 hours of BioBlitzing. But all hands were on deck for the BioBlitz, with each DR group contributing to the day.
One of the expert-led collections was freshwater-invertebrates, which involved us getting hands-on in the creek. We found shrimp, hydra, snails, and so much more. The rest of our SMS student group alternated between hosting talks for children, showcasing the ants and the dung beetles under the microscopes, and the other groups were able to go to expert-led talks.
So overall, we found 235 unique plant species, 81 unique invertebrate taxa, 60 of which were insects, including the 9 moths that we sampled during the light sheeting event, and 69 unique vertebrate taxa, including the Lumholtz tree kangaroo, the spectacled flying fox, and the green-eyed tree frog, all 3 of which are species of special conservation interest in the wet tropics region. And finally, as you can see, there's a good number of biodiversity in this revegetated forest already, but as the understory continues to develop, the canopy continues to thicken, we will only expect that biodiversity to increase.
We collected a total of 50 specimens with a variety of moths, butterflies, spiders, and leaf litter invertebrates. Our specimens were then taken and analyzed under the microscopes and posted on iNaturalist. Some of the specimens, like the microspiders, need microscopic analysis to get done at species level.
The question often gets asked, haven't we passed the period where we pin insects up? And the reality is no. If you look at moths, there's somewhere between 20 and 30,000 species of moths in Australia, many of which we don't even know exist yet, and a lot more that exist in the National Insect Collection just as a number. So there's still huge amounts of work to discover the biodiversity we have in Australia. If you've got something that's really endangered, governments will not put money into species unless they're named. So if you have a species that has no name, it's not going to get any particular funding. The cornerstone of biology is actually knowing what species are, their life histories, and how they fit into the more general ecosystem. And without knowing that, we don't have any idea of what make ecosystems tick, and without knowing that, we really don't know how to help look after them.
So at the School for Field Studies, we're currently setting up a natural history collection of plants and animals with the hopes that this would be a useful resource as it grows for students and future researchers who come to visit the site and want to study the biodiversity of the region. So all the collections that were collected in the Malanda BioBlitz is going to be curated in this natural history museum that we are currently setting up.
Besides engaging to biodiversity, we watch the community getting together to learn about nature. The BioBlitz is far more than just numbers of specimens that you collect, and the total biodiversity is about people reconnecting to nature around them.
Let people know that there's actually, high biodiversity, lots of species that live in just these urban environments or revegetation areas.
Special thanks to our peers in the community for participating. You made the BioBlitz a BioBlast.